Hello and welcome to my channel Omni Bioentrance, and today we will be discussing IIT JAM question paper for the subject Biotechnology conducted in the year 2020. If you want still previous year question papers of IIT JAM, you can go to my channel Omni Bioentrance. In the channel, see for the channel playlist. In that you will find IIT JAM, where you can find question papers which have been solved with explanations. If you are new to my channel, please do like, share and subscribe. Coming to the first question. Deficiency of the enzyme phenylalanine hydroxylase causes phenylketonuria. Yes, that is correct. Phenylalanine hydrolase converts phenylalanine to. So it catalyzes the hydroxylation of phenylalanine to tyrosine. It uses molecular oxygen and tetrahydrobiopterin as cofactors. So answer here is option C, that is tyrosine. T and B cells they are called as lymphocytes. Answer is T lymphocyte, B lymphocyte. Answer is option A. In humans, the testis temperature is maintained below the core body temperature with the help of scrotum. Answer is option C. Then next one. Protein that helps other protein to fold correctly are chaperones. Answer is option A. Proteasome, it is a complex that degrades misfolded proteins. And ubiquitin, it is a protein tag and it marks the protein for degradation. Desmosome is a cell adhesion complex and it is not related with protein folding. So the answer here is option A, that is chaperone. The correct sequence of phases during mitosis. Okay. Prophase, metaphase, anaphase, telophase. A itself is the correct answer. In viruses, capsid are made up of. So capsid in viruses, they are made of proteins. Answer is option A. Then the common component in crustacean exoskeleton and fungal cell wall is. So fungal cell wall, it is made of mainly chitin, and crustacean exoskeleton is also of chitin. So that is the common component. So answer is option C. The correct sequence of evolution, simplest to the complexes. So the simplest are the algae, then comes your bryophytes, then pteridophytes, then gymnosperms, and then finally angiosperms. So if you see the options here, you can see it is option A, that is algae, bryophytes, ferns. Ferns belong to pteridophytes, then angiosperms. So answer here is option A. During El Nino, cold water of the north flowing Peru current is displaced by low nutrient warm southward current. A itself is the answer. Then next one. Match the deficiency condition in group 1 with the corresponding vitamin in group 2. Beriberi is due to thiamine deficiency, vitamin B1, P3. Scurvy is for ascorbic acid, which is vitamin C. Birth defects are for folic acid 4, and night blindness is because of retinol, S2. So the answer here is option C, that is P3, Q1, R4 and S2. Then eutrophication refers to an aging process from A. So eutrophication, here the body of water or the water body, it becomes enriched with excessive nutrients. So that process is called as eutrophication. And because of eutrophication, you will have excessive plant growth, that is algal bloom. And there will be depletion of dissolved oxygen, and it can harm or it can lead to death of aquatic life. So here if you see, low production ecosystem to high production ecosystem due to availability of excess nutrients. That is correct. A is correct. Then next one. Match the enzymes in group 1 with corresponding substrate in group 2. Amylase. Amylase is starch, P4. Pepsin is protein, Q1. Lipase is fat, R2. And ribozyme is RNA. It is catalytic RNA molecule, ribozyme. So S3. So the answer here is P4, Q1, R2, S3. That is option B. Then next one. In ABO blood grouping testing, which of the following is incorrect? Now, if you see A group, agglutination with anti-A antibodies. So, A blood group, it has got antigen A and antibody anti-B. It doesn't have antibody anti-A. So, A is the antigen. So, when you add anti-A antibodies, it will result in agglutination. So, A group, A option is correct. Now, B group, agglutination with anti-B antibodies. Now, B group, it is having antigen B but no antibody anti-B. That is not present. So antigen, when you add when you add anti-B antibodies to B, antigen B will react and it will result in agglutination. So B option is correct. AB group. AB group, it has got antigen A and antigen B but it has no anti-A and anti-B antibodies. So they have mentioned, you have to see for the incorrect statement. So no agglutination with either anti-A or anti-B antibodies. How can that be? There will be agglutination because AB is having both antigen A and antigen B. And when you add anti-A or anti-B antibodies, there will be agglutination taking place. So C is not the correct answer. O group, if you see, no agglutination with either anti-A or anti-B antibodies. It doesn't have antigen A and antigen B. So naturally there will be no agglutination. So D is the also correct. Now what they have asked is which of the following is incorrect. Now C is the incorrect option. Answer is option C. Then next one, match the RNAs in group 1 with their corresponding function in group 2. snRNA. snRNA, it is for RNA splicing. P2. piRNA is for germline protection from transposable elements. snORNA is for rRNA processing and siRNA is for selected mRNA degradation, that is coming under RNA interference. So answer here is option D, that is P2, Q4, R1 and S3. Then next one. In a typical mammalian cell, the protein content is 20% of its net weight. If the density and volume of the cell are so and so respectively, then the concentration of the protein is. So they have given the cell weight here, protein weight, protein concentration, density they have given, that is 1.2 grams per ml, volume is 4 into 10 raised to minus 9 ml and protein percentage is 20% or 0.2. Now you have to calculate the cell weight. So cell weight is equal to density into volume, because density is weight by volume. So cell weight will be density into volume. So 1.2 into 4 into 10 raised to 9, that will give you 4.8 into 10 raised to minus 9 grams. Now protein weight you have to calculate, because protein concentration you should know the protein weight by volume. Then only you will get the concentration of the protein. So protein weight is cell weight into protein percentage. So 4.8 into 10 raised to minus 9 into 0.2, that will give you 9.6 into 10 raised to minus 7 milligrams. The answer is in milligrams per ml. So now put it in the formula for protein concentration, that is protein weight by volume. You will get it as 240 milligrams per ml. So the answer here is option D. Then next one. Match the type of bacterial flagella in group 1 with their definitions in group 2. Monotrichus is only a single one. So P4, a single polar flagellum, that is monotrichus. Peritrichus is all over the body of the bacteria. Or it is the flagella distributed over the entire cell. Q3. Lophotrichus is two or more flagella at one pole of the cell, and Amphitrichus is flagella only at both the poles of the cells. So the answer here is option B, that is P4, Q3, R2, S1. Then next one. Match the disease in group 1 with their causative organism in group 2. Now syphilis. Syphilis is caused by Treponema pallidum, P3. Then bacterial dysentery by Shigella dysenteriae, gas gangrene by Clostridium perfringens, whooping cough by Bordetella pertussis. So the answer here is option D, that is P3, Q1, R4, S2. Then next one. Determine the correctness or otherwise of the following assertion and reason. Assertion. The difference in the respective melting points of butter and coconut milk is caused by degree of saturation, of course, of the corresponding fatty acid chain. That is 100% correct. Then reason. Unsaturated fatty acid chain in fats and lipids become solid more easily because they are relatively straight and thus able to pack together more closely than saturated chain. That is not at all correct, because unsaturated fatty acid chains, they have one or more double bonds between the carbon atoms, and this can result in kinks or bend in the chain due to this double bond character. So they cannot pack tightly just like the saturated ones, and they remain liquid. Because of these reasons, they remain liquid or semi-liquid at room temperature. Okay, so that is the case with unsaturated fatty chains. So here reason is not correct, but assertion is correct. So answer is D. A is true but R is false. So what will be the correct statement for the reason is unsaturated fatty acid chains in fats and liquids, or fats or liquids, become liquid more easily because they are relatively bent or kinked, making it harder for them to pack together closely. Okay, so A is true, R is false. Match the techniques in group 1 with their application in group 2 for protein analysis. Nuclear magnetic resonance spectroscopy. It is for structured NMR, protein structure, P4. P4 is there only in one option, so B itself is the correct answer. Fluorescence resonance energy transfer, FRET. It is for proximity between specific sites. Ultraviolet absorption spectroscopy, UV spectroscopy. R2, it is for concentration determination, that is concentration of proteins. Biuret test, Lowry test, all these depend on UV absorption spectroscopy. Dynamic light scattering, it is for size determination. So P4, Q1, R2 and S3 is the answer. Now determine the correctness or otherwise of the following assertion and the reason. Assertion. Lac operon is an inducible operon. Yes. Why is lac operon an inducible? Because its expression is triggered by the presence of an inducer molecule. That is your lactose. So lac operon is an inducible operon. Then reason. Lac operon is not induced when the repressor protein remains bound to operator DNA sequence. That is also correct. But it doesn't tell why the lac operon is called an inducible operon. Okay. So here both A and R are true, but R is not the correct reason for A. Answer is option B.